Welcome, everyone, and thank you for coming zooming in for this webinar that is sponsored by Gratiot Lake Conservancy. We're really happy to have Dr. John Younger, with us tonight. And John, I've known John for I think over a decade now, and he's been involved with the Conservancy for quite some time and doing studies there and collecting data since 2015. John is a professor at Governor State University in Illinois, and he's a biology professor, and his specialties are population ecology, vertebrate zoology, and conservation biology. He and his wife Linda and their son have a camp in Gay up in the Keweenaw. So he's got a real deep connection to the area, even though he doesn't live there all year. And John has been working with small mammal populations, both at Gratiot Lake Conservancy and Rabbit Island. And tonight's talk will be about the data he has collected and some of the conclusions, or at least hunches he has now about what's really happening with population dynamics between these populations.
Okay, I'd like to thank Bonnie for the invitation for presenting today, and looking forward to some conversations maybe after getting through this talk. And again, also thank GLC in general for access to the land, as you'll see in a while. And as Bonnie's already mentioned, going to be discussing mammalian predator-prey dynamics, temporal trends, and a predator-free island. Promise not to spend too much time on this here, but give a little bit of the quantitative background in ecology. Ecology is rich in models, and some of the original predator-prey models, such as Locke to Voltaire's predator-prey equations, go back actually almost 100 years now, and Holling's models go back over half a century. Very, very briefly, these are differential equations. For example, with the Locke to Voltaire, are you able to see my cursor when it moves okay? Okay. D, or delta, is the change in predators, and that is a change in predators over time. H is denoted as prey here. So essentially what these equations are doing is multiplying how fast they can increase, the rate of increase R, times the number of individuals. And here on this lower one, they are subtracting off alpha, which is the effect of the predators on the prey, specifically how much they're eating them. If they consume them very rapidly, then the change in prey over time goes down. And a big question in ecology has been how well these mathematical models fit what is happening in the real world. And that requires empirical data then for testing these models. And the theoretical folks are constantly developing new ones to be tested, probably at a greater rate than we have data to test them with. This has also led into numerous concepts, and these are again models that could just be explained verbally. A hot debate in ecology for 30 years has been top down versus bottom up. Top down would be organisms higher in the food chain are going to reduce or limit the number of prey below them. Lower trophic levels is how it's generally referred to as. For example, sharks, or even more appropriately, killer whales. Killer whales can limit sea otters. Sea otters influence urchins. Urchins influence kelp. So if you remove killer whales from the system off the coast of California, then the kelp beds are going to go up. Or is it a bottom-up system, such as on the right, where kelp production is going to influence the numbers of the tertiary, top, or final predators? Taking it onto the land here, this is on the left-hand side, just graphically depicting these ideas. And this is taking it one step further. Weather. Rainfall makes a big difference in plant growth. And there's a way I'll mention later on if there's time NASA has a satellite up that will actually calculate what's called NDVI, the Normalized Difference Vegetation Index, and it can determine how fast the plants are growing. And it is almost always linked to rainfall levels. So good or bad these days, you can just sit in your office or your lab and actually measure off the satellite plant growth rates. The plant growth rates then, as depicted in this figure, are showing more food, snowshoe hares go up, voles go up, ptarmigan numbers go up. As they increase, that provides more food for the top predators. In this case, we're just using golden eagle. On the right-hand side, classic example, the snowshoe hare lynx dynamics from Canada. And there's data on those back to the 1700s due to fur trapping. And the original assumption was that the lynx are limiting snowshoe hare numbers. There's been some experimental work in the Yukon, though, where they provided supplemental food to the hares, and the hare numbers actually increased dramatically. So another idea here is that you could actually merge together top down, bottom up, and they play an interacting type of role. In other words, food availability makes a difference in the hare numbers. As you could see here during the winter, when they're starving, they'll be eating the bark. And during the winter months also, their numbers could go down from lynx, so they could be interacting. And if food becomes very scarce, that in turn then would actually, let me step back here. If the plant food becomes very scarce, the hare numbers can decline, and then the hare could be indirectly influencing the lynx or the predator population. So we've got extensive empirical data. However, an unfortunate reality, depends on who you ask, is that a lot of ecological work gets done in units of 1 to 3 years. Our graduate students want to actually graduate, and the typical master student is spending 1 to 2 seasons in the field. Typical PhD student is spending 2, maybe 3 seasons in the field. So very often projects are limited to timescales. They can also be limited to funding if there's going to be extensive work. Same applies to these spatial scales. Working in a woodland such as this is not that challenging, really. You scale it up a little bit, it becomes more work. You scale it up to the landscape here on the bottom, and it even becomes more work yet. And I'd suggest that handling the larger spatial scales is a little more straightforward than time. You can get large field crews, classes together, what have you, and spread them out across the landscape. Keeping people around, though, for years becomes a bigger challenge. So I would strongly emphasize there is a need for long-term studies. Now, that said, there are many of them. The National Science Foundation has what is called the LTERB, or the LTERs actually, long-term ecological research sites. So these do exist around the country. This paper was actually just published, actually just came out about a month ago in a journal, Ecology, and it cites, or showing here, 6 different long-term studies of small mammals around the world. And there's a few others. There's a very well-known site in coastal Chile. There's another one in Ontario. And these illustrate the need, in my opinion, for why we need these extensive databases. We'll take a look here at the Kazakhstan one. And this is with the greater gerbils in Kazakhstan. If we just pick any period in here, say jump in the mid-70s, take that to the mid-80s, about a 10-year time span. Even one 10-year study, in this case, all that's going to tell you is that the individuals went up dramatically, went down dramatically. And in the big picture, that's really not answering ecological questions on population dynamics and what's determining the number of individuals. Myself, many ecologists also would probably suggest you need a minimum of 3 repeating patterns before we start to believe it. So if you go back into the 60s, take this up into the 90s, you could see 3 of these cycles. I begin to believe it at that point that they're fluctuating on a 10-year cyclic basis. But again, you're talking 30 years, which is many people's careers before you actually start to see these patterns completed. And this assumes that a government agency, a new prof, what have you, has the resources available to start these studies up, which is rarely the case. Again, our National Science Foundation is one route, or some of these studies are being handed off to graduate students or colleagues that are younger to keep these going. But again, it's limited, and it does show the need for the extensive temporal scales. So 3 broad questions going to be addressing here today, and that much of this work was originally set up to look at. Do predator populations drive prey population dynamics? So that would be assuming a top-down model. Are small mammal population dynamics synchronous? And this has again been debated, population cycles, synchronicity in populations. It appears to be latitudinal in variation. Once you get far north, say into the Arctic, many of these animals appear to fluctuate on a 10-year or a 3 to 4 year cycle. And this includes some of the birds too. Ptarmigan, for example, the lemmings, the predators. At more southern latitudes, say 30 degrees, these patterns do not appear at all, and they are much more asynchronous. In northern coniferous hardwood forests, though, there's very little data. And the Keweenaw falls right into this area at around 47 degrees. So this whole area of about 45 to 50 degrees north latitude, there's a paucity of information here to describe the synchronicity or asynchronicity that may be occurring. And how does the absence of predators alter small mammal demography? And again, there's multiple ways to go about doing this. You can do it experimentally and exclude them. That's happened a few times. It is a lot of time, money, and work. And so far, it's only been done on relatively small scales. They have removed predators, which gets into a huge ethical debate where you basically go out and shoot them is what it boils down to. And it's effective over large spatial scales, but does raise, obviously, other issues with the management practices. A third way that it's been done is islands. And one of the best known long-term studies is, again, very close to home in the Keweenaw, the Wolf Moose Isle Royal Project. And these are slides that I think everybody's familiar with. When you're at national meetings or international meetings, it's standard operating procedure to show these, but I think most everybody knows these pretty well. GLC is obviously located right up here. There's the lake itself. And Travers or Rabbit Island is located out here, Rabbit Bay. When I started looking into this back in 2012, I had 2 broad criteria that I wanted to try to meet. One is what is referred to as site matching, that the sites would be broadly, actually more than broadly, relatively close in their habitats. The second is long-term stability. I'll give you an example here. If you've got Rabbit Island here, one of the areas I looked at is Travers Point. When I looked at it in 2012, it looked perfect. It was about 40-year-old forest, so it probably would have worked. In 2013, the logging company started on. So there's a limited number of areas where you can access with that stability. So GLC has been great for that. It's been decades since it's been cut, which helps it match up better with Rabbit Island. Excuse me. Also, the geology of the 2 is broadly similar. Even though it's close to the rift up here, it is still on the Jacobsville sandstone, which the island is also on. And the geological history does play a big role in influencing the habitats and vegetation in a particular location. So here is Rabbit Island itself. The red line up here from the mainland, that is approximately 2 miles. So the minimal distance you're looking at there is 2. There's cliffs out there, and it would require about a 20-foot dive from the critters, which we're not going to get into today. I'll focus more on the GLC than on the island. And to give you a feel for the island, that red line here is about half a mile in length. And here's some of the hardwoods on the interior. Mainland site, again, GLC property. running up through this area. The cabin field station itself is right about down here. That's Beaver Pond right there. Just some sites of the interior, again, mostly maple hardwood, and that transitions into coniferous forest with hemlock and cedar as you head a little bit south. Keweenaw is also very well known, or at least is recognized for a very high diversity and abundance of predators. So if you're interested in asking questions on predation, that lends itself to those topics very well. Again, not going to, due to time, get into all of these. If anybody wants to look at them more carefully later on, they can. Some of these predators are common in the area, some are not. Some play a big role in small mammals, others do not. For example, raccoons are highly omnivorous. Both foxes go after small mammals, deer a little more on the wolf side. Again, many mustelids in the area. Mink are a little bit more aquatic, but long-tailed, least, ermine feed very heavily on the small mammals. And two that I'm going to hit in particular later on, fisher and pine marten. Okay. Remote cameras. Remote cameras are very well. We have a dozen of them out at GLC. There's pros and cons to these. They are low maintenance, low labor, and they can bring in a lot of numbers. The disadvantage is you cannot generally identify individuals. So for at least ecological work, you can't quantify how many are present. You can say that if you see a bear, you could have one bear seen on 10 nights or 10 times in one night, or you could have 10 bears seen over 10 nights. And it's hard to make that call. Lower left here is probably a red fox. The body to leg length ratio suggests red fox over gray fox. Lower right down here, that's one of our pine martens. It is checking out one of the Sherman traps that I'll talk about a little bit later on. And the bear on the top is checking out one of the pine marten traps. Started doing some camera trap work in 2013 at GLC just to get a feel for what predators were around there. And hands down, what showed up on the images most were black bear and the pine marten. And that's continued to this day. I was out there 2 weeks ago getting the cameras out for the spring, and I'll take a guess, same as in the past years, those two would be the most abundant. Bear are very omnivorous. They eat far more plant material than animal material, so chose not to focus on the bear very much. Pine marten, depending on where you look at, are possibly lightly omnivorous to what is referred to as a hypercarnivore, meaning they feed almost exclusively on meat. And because they showed up on the camera so much and their hypercarnivory, we chose to focus on the pine marten as the main predator at GLC. Again, around GLC, there's a lot of others. Gina, Mark, they've got cameras scattered over the whole area. And they're actually getting more pictures of fisher and wolves in some of the surrounding habitats. Some of the mammals then, the smaller mammals here, the prey items. This is what we refer to as a trapping grid. It's just a bunch of points. We try to get these as equally spaced as we can based on the GPS coordinates. These are probably within 1 meter. It is referred to as an 8x8 array. So 8 rows, 8 columns. That gives you 64 traps. It is 12 meters between each trap. And we assume that it is sampling half the distance between each trap. So that gives roughly a 96 square meter area, 96 by 96 meter area, which is getting close to 10,000 square meters or approximately 1 hectare area that it's sampling. And there are 3 of these grids at GLC and 1 on the island. Each of these grid points are flagged. That orange flag there, and actually you can barely see it in this image. There's one of the Sherman traps in the back at GLC. This is out on the island here, and we have to use rebar out there. The island gets a little more rocky, and so these give a more permanent fixture. Diverging very slightly, one difference in vegetation between the two is hemlock. It is really interesting seeing the effects of deer browse on some of the plant species. As I'm going to guess many of you are aware, very little, excuse me, ewes, not hemlock, ewes. Ewes in the Keweenaw are not common. You can walk around GLC and some of the areas, it's almost like parkland moving through it. Ewes on Rabbit Island range from about 4 to 8 feet in height, and we almost have to flail our bodies into it just to push through it. It gets so dense in areas. And again, that's presumably deer browse. And at each of these here in the lower right, that's a Sherman trap. Again, with the flag point, that one's actually in Illinois, not in the UP. We process each of these animals and just grab them in a little bag, and we can get the data that we need off of them. Each trap is baited with peanut butter and rolled oats. For some reason, a peanut butter rolled oat mix works excellent the world over. Used it in multiple continents, and critters love it. Matter of fact, Gina was just working with someone. They were trying to get Fisher up in the, no, they were trying to get boreal chickadees to come to peanut butter up in the trees, and the Fisher were actually climbing the trees going after the peanut butter faster than the birds got it. We also provision them with an apple slice to help reduce dehydration, and we put polyester bedding into them. We do this in the fall months, and again, October, we've had it snow on us. So that helps keep the animals warm in these metal boxes. Each individual receives a uniquely numbered ear tag so that we can track them and keep count of them. And data is recorded on the grid point, the species, its gender, its mass or how much it weighs, and its reproductive status. We also have at times collected feces for parasite analysis, and we'll sometimes take a little snippet of ear tissue for potential genetic analysis. Group of students, there's the traps. Good experience for them, man. Good labor for us. And they're out working on some of the grids. And here's, I think that's actually Linda, checking one of the traps down the cedar swamps. Squirrel trapping. So in addition to the Shermans were used for mice, voles, mostly flying squirrels, red squirrels and chipmunks. We have larger cage traps out for them. These are in a 5x5 array, 25-meter spacing, so 25 traps per grid. And then we also have the marten trapping going on. In the upper right, you can see a pine marten. Same picture in the upper left. We cover these traps up since they're mesh. If it rains, due to the long, narrow body of the pine martens, we don't want them getting wet and risking hypothermia with them. Plus, it appears to help increase trap success. They like to go down these narrow little tunnels into the traps themselves. Traps get baited with a rat, and they're lured with a commercial marten lure. So the lure draws them in, and to the area, the bait brings them into the trap. These dotted areas, these three, represent each of the 3 8x8 array small mammal trapping grids at GLC. So again, each of these areas with the small dots, you're looking at about 100 meters across these. The squares represent the pine marten traps. So the pine marten grid is a 4x8 array with 100-meter spacing. So you're looking at it, the whole area is about 1 kilometer in length and about 0.5 kilometer wide. It's a little less, but we're sampling about 0.5 square kilometer for the pine marten, which labor-wise is moderate, but in terms of the pine marten, with how rapidly they can move, it really isn't that large of an area. And more recently, have started seed traps. This is one thing I wish I would have started initially with this project. Hindsight is always easy to go, I should have done this right from the beginning, but the traps are 5-gallon buckets, that's in the green. There's a brick in the bottom that keeps them from getting rolled over, holes drilled in there for draining, and this is aluminum flashing cone. It samples a 1 square meter area, and this prevents any of the mammals from climbing up into it. And then there's a mesh bag at the bottom, so as the seeds drop in, it catches them in the mesh bag, but the rain can pass through, go out through the holes and not rot the seeds up. So I just put these out again a couple weeks ago, and I'll probably check them once mid-season or so to make sure everything's working okay. And again, seed traps, sugar maple up here, red oak, hop hornbeam, ironwood, depending what common name you go with it, cedar, and hemlock, all potentials there. One thing that we have found is that the hop hornbeam is probably more of an important food than I would have ever have guessed. And many of the squirrels seem to go after that relatively heavily. Data analysis, just going to touch on this very briefly in here. It is a very important topic. Okay. Data analysis, statistics generally assume an independence of samples. This is both in space and time. This spatial temporal work, they're not independent. What's referred to as autocorrelation. And this is actually important. What happens at time t, say in 2020, influences what happens at time t+1 in 2021. So we need to take that into account, how the number of individuals this year are affecting the number of individuals next year, and maybe even 2 to 3 years into the future. That's one of the questions that can be interesting with the synchronicity. And we analyze these data with a technique called repeated measures analysis of variance. It is a modeling technique that is designed to take into account autocorrelation. I'm not going to be getting into it anywhere beyond this, but at times I will mention these are significant or not significant, and it's in reference to the statistical analysis of the data. Okay, here's Rabbit Island. And again, not going to get too in depth on this. We now are getting into about 10 years of data from the island. As you can see, there have been many species that have shown. Rabbit Island, again, can give explanations for each of these, but I'll keep it brief, won't get into too in depth with how we know how these different mammals are out there. It is not quantitative. Some of it is haphazard sampling. But the one take-home message here that will relate to GLC is the redback vole. As you can see, others have shown up and disappeared, and the only one that has been persistent in relatively moderate, and it turns out in high numbers, is the redback or Myotis gapperi. Here is for the mainland results then. 4 main species are the key players here. The redback vole, Myotis gapperi, American red squirrel, Tamias cyrus adzonicus, woodland deer mouse, Permiscus leucopus, and the eastern chipmunk, Tamias striatus. Several others have shown up, but they are not nearly as common. White-footed mice are now appearing. Historically, they've not been in the area. They appear to be displacing the woodland deer mouse. Northern flying squirrels are the indigenous species. Southerns are showing up at GLC. Actually, they're more common. The southerns now appear to be replacing the northern flying squirrels. First gray squirrel was seen out there last year, and 3 species of shrews have also been recorded at GLC. But their numbers are low enough that data analysis is challenging. Okay, going to get into some of the data here now, and these are the graphs. Some of this can get a little tedious working through these. These are the 4 main species that I was mentioning a little while ago. And not major surprise, initially back in 2016 is when we had the highest abundance, and that was with Myotis and Permiscus. These are average numbers for the 3 grids. So that would mean between the 3 grids, you're looking at roughly around 90, you're getting upwards near 100 individuals that are tagged across the 3 sites. And rapid decline going into 2017. They've stayed relatively low with the exception of the chipmunks. And again, many of you are probably aware, last year seemed to be the year of the chipmunks throughout much of the peninsula. Their numbers actually doubled, almost tripled compared to some of the past couple of years. Okay, this is now overlaying the pine marten with the prey. So the blue here are the marten numbers. These in some cases are exceedingly high pine marten numbers. If you look at this one here, that is 14 individuals in just less than half a hectare or half a square kilometer. So you're talking about 30 pine marten per kilometer, which is the highest that I've been able to find in the literature yet. So GLC is supporting an extremely high abundance of these predators. Their numbers obviously fluctuate. Many years they've dropped down to just around 3 or 4. This is going to illustrate some of the need to see long-term and why I still have to be careful with some statements. If you only looked up through 2019, the numbers on these statistically were relatively synchronous. When the data was reanalyzed, though, going into 2020, this divergence of the chipmunks was enough to make their dynamics considered statistically asynchronous, meaning that they are not fluctuating the same. And we'll see if that continues as we go into the future. Also, up until 2017, my guess was that a large number of the small mammals here were driving this increase in the pine martens. And I'll talk about that more in just a few moments. So another way to help clean these graphs up, as opposed to having to look at 5 separate lines, which can be scattered and hard for the human eye to see the patterns or trends, is to take the total number of prey. And this represents the 4 common species here in the orange, and relate that then to the pine marten numbers. And again, you can see here when they peaked, the marten went up, again suggesting that the number of prey might be playing a role in driving pine marten abundance. When the prey crashed, pine marten were relatively low. We'll see what happens here with the total number going up. I will be very curious if starting this fall, we see a slight increase again in the pine marten numbers. That would be again suggestive, not of top down, but of bottom up, where the number of prey at GLC are directly influencing the numbers of the main predator. This is a very messy graph. It's not yet done. I decided to toss this in at the last minute. One problem with comparing the total number of prey to the predators is that all prey are not created equal. If a marten catches one vole, it's getting about 24 grams of biomass out of that, and a lot of that's water. If it captures a red squirrel, it's getting about 7 times that or 8 times that. You're looking at closer to 200 grams. So what I've started to do is take the total biomass of the prey and try to then relate that to the predators. One of the problems here is that if you use the same scale, that's why there's not this axis is not labeled. The blue line is prey biomass, and the orange line is the number of pine martens times 1,000. That may seem silly, but to get them to overlay on each other, it requires multiplying the martens by such a large number. When you run a correlation analysis on this, turns out that there is a perfect inverse correlation between prey biomass, the actual volume of food out there, and how many pine marten you have. And we will see what happens then going next year. Is it more the number of individuals? In that case, pine marten should probably increase. Or is it biomass? In which case we will probably see the pine marten slightly drop because the biomass is not extremely high here. And to bring in a little bit of the comparisons with the island, these are population dynamics of myotes or the redback voles on Rabbit Island. As you can see, they are higher than almost any time at GLC, and they stay very stable. 2 potential explanations. Islands are great, they're big, they're realistic, but they're not controlled well from an experimental perspective. So a second explanation to predators is that it could be competition. In other words, You saw that table that showed the other small mammals on Rabbit Island. The voles basically have no competition out there. I still have a strong hunch, though, that it is predators. And in the absence of predators, the voles are averaging about 3 to 4 times greater density, and their dynamics are very stable. So they're not being driven up and down very much. And comparing that to the mainland site, you could see during 2017, the voles almost disappeared completely. And you don't see anything like that happen. Now, again, question remains, are these fluctuations on the mainland due to competition with other small mammals, or is it the predators influencing them? So that is still some of the questions that need to be refined better, or actually the answers need to be refined better. So some broad conclusions here. Do predator populations drive prey population dynamics top down? It's appearing at GLC, at least initially, probably not. Prey may drive the Martin numbers, which would suggest a bottom-up system. Our small mammal population dynamics are synchronous, and they appear to be asynchronous, at least at 47 degrees latitude. Statistically, at this point, they are asynchronous, but we look at what are called p-values, and the p-value is just barely 0.05. So if you had to draw a conclusion now, it's asynchronous, but not strongly. And how does the absence of predators alter small mammal demography? At least based on the one species that has consistently been at the island, there is no question they have greater abundance and increased stability. The greater abundance is almost threefold. But again, we have to put the caveat of competition in there. Possible future work. Interested in incorporating NDVI data, and actually NDVI, Normalized Data Vegetation Index. This is what I had mentioned earlier that NASA data has, and they now have it down to 30 by 30 meter resolution. So it would be possible to take what are referred to as pixels, and with 30 by 30 meter resolution, overlay 9 pixels on each of the 3 grids on the mainland and the one on the island, and could see how fluctuations in primary productivity might be influencing their numbers. And this on the right is just a graph that NASA, I believe this is a NASA generated one, that shows primary productivity global off of their data. The other thing that I've been interested in quite a while, hasn't happened, is looking at genetic divergence of the voles on the island compared to the mainland. New grad students probably starting in the fall that is interested in genetics, and we just hired a conservation geneticist here at GSU. So I'm optimistic now that this will be moving forward in the next year or so. And talking with Rob Gorski, we've got some ideas on how they may have gotten to the island. We'll see if the genetic divergence times correspond with some of the folklore from the area, which that can actually be quite useful trying to merge the two together.